Okay, let's get started. Thank you.
Okay, so our project is the investigation of biologically active cythine diterpenoid production in Hericium abietis. And before we start, we'd like to thank those involved, especially our professors Paul Prispilowicz for providing us with the strains, Lolita Calabria for helping us along the whole process, and also Anna Barim up at Washington State University for helping us out with the analytics. So we wanted to start off just by looking at a general overview of the Hericium genus. It is in the order Russellales, along with the genus Ruffula and Lactarius. There is estimated to be 23 species worldwide, 4 of those growing in America, and 2 of them are, as you see on your right, Hericium abietis and Hericium erinaceus, and they are all white rot saprotrophs. Hericium abietis is branched with spines and favors conifer trees. And what's interesting, and the whole reason why we did this project, is because it has never been formally researched for the bioactive compounds. And Hericium erinaceus, otherwise known as Lionsmane, grows in a clump or clumps with spines, and it grows on hardwoods. And it is the most studied species by far in the genus and has a lot of promising research behind it, which leads us to our first question. Does the mycelium of Hericium abietis contain cythine diterpenoids? And we also wanted to explore, how does the cythine diterpenoid profile differ between abietis and erinaceus when they are grown in the same media? You're on mute, Jax.
I just saw that. Thank you. So you're probably wondering, what are cythine diterpenoids? Well, they're defined by this tricyclic ring structure you can see here with the cursor, where we have a 7, a 5, or a 7, a 6, and a 5 carbons in a ring that are in this bent ring, and that is becoming a common shape in these different classes of molecules found in fungi to have bioactivity. And out of the cythine diterpenoids, there's over 170 that have been found, and they're all secondary metabolites specific to the mycelium of Basidiomycetes. Basidiomycetes are the species or the phylum of fungi that are the ones like with caps and gills that we most often recognize, not as much of the amorphous shapes. And cythine diterpenoids were originally found in the Cyathus heleni fungi, and that's why it was named cythine after that genus. So some of the bioactive properties of these compounds include nerve growth factor promoting, neuroprotective, antimicrobial, anti-MRSA, anti-inflammatory, and others. And you can see it in bold here. Erinacine is the one that we're going after for research in this particular mushroom or mycelium of the fungus. So Erinacine A has been hotly researched in the last 10 years or so for its potential treatment in neurodegenerative disorders. And this is because these cythine diterpenoids stimulate production of nerve growth factor in the body. And so in vitro, it's been found to promote the neurogenesis in these cell lines that show that it has potential for neuri outgrowth in animal models. So then there's been quite a few animal models showing the mechanisms as well as the different effects. And there's one significant study showing that it delays cognitive decline in aging mice. And in human trials, we've even seen it to increase cognitive performance in Alzheimer's patients.
Jax, I'm just pausing real quick. We have a couple more in the waiting room. I just got a text, and so maybe we could do both. throw them in real quick.
Thank you. Yeah, it's not popping up for me now that I'm in slideshow mode. I appreciate that. So I have to like take it out of this to see the waiting room.
Quick little.
There we go. Thank you. Is there someone else? Yeah, you'll have to tell me each time because I have to stop the slideshow to get to it.
I think that's it.
Yeah, if someone else
Hey,
comes in,
gentlemen.
just let me know. Thanks. Hey, welcome.
Yeah.
All right. So where were we on slide? So some of the H. erinaceus or Hericium erinaceus, the Lionsmane bioactive compounds, include a whole slew of erinacines and many other polysaccharides, and you can see I put a hericinone here at the bottom, which is a benzaldehyde. And so we're just limiting our focus and our scope to erinacines in particular, these type of cythine diterpenoids. But I put a few different compounds up here so you can see there's sometimes some difference in for example, in hericinone, they're found in the fruit body of the mushroom and not in the mycelium. And it's a little debated in the literature whether it actually produces neurogenesis or if it's just neuroprotective. But Erinacine A is by far the most studied compound out of the group, and maybe within the whole the fungus. And so there's anti-tumor, anti-inflammatory, neuroprotective, and others that have been found with this molecule in particular.
Oh my gosh, you guys.
So some of the effects of Erinacine A, I put a few up here that have that drew my interest, some of the more recent studies. And one of the older studies is just showing that it's a much more potent stimulator of nerve growth factor than the positive control epinephrine. And this is in animal models. And we've also seen it increase the level of myelin basic protein on neuronal fibers and increased expression of oligodendrocytes in the corpus callosum of rats. Now, I'll break that down a little bit and why that's important. Myelin and the basic protein for myelin that becomes myelin sheathing is an insulation around your nerves. And it's not known that we can really regenerate this and damage. And this is why diseases like multiple sclerosis are so awful for the symptomatic decline, because when the myelin sheath degrades, we don't really have the potential to grow that back naturally. So there's very robust interest in a medication that could potentially do that, and many research dollars are poured into that each year. So this is one example of a potential treatment. We also see here that there's actually it prevents lipopolysaccharide mediate glial cell activation to protect dopaminergic neurons against inflammatory factor induced cell death. And that's in vitro and in vivo. And what that means is inflammation in the body is one of the most significant markers for disease in many types of disease, especially in the nervous system. And to have and cell death is commonly the cause of those symptoms in the nervous system inflammation, and this is a constant problem in neurodegenerative disorders, but many other disorders and even acute traumas as well, like head traumas. And so this is actually a very significant finding, to be able to protect your dopaminergic neurons from death, basically, like say during a stroke or a head trauma. And that's what this is, what there's the most interest for. And we also see that over time, we can stimulate nerve growth gene expression and nerve growth protein transcription. And this also enhances nerve growth factor secretion from the glial cells. And that one I'd like to break down just a little bit further so you understand just how important Lionsmane can be. And I think this is probably one of the most obvious reasons why people say, I take Lionsmane and it works. For example, I was just at the store and I was talking to the checkout person about this presentation because they had a mushroom sweatshirt on, and and we got into a conversation about mushrooms, and she's vegan and she loves mushrooms. It's one of her favorite foods. And so I asked her about lion's mane. She said, yes, I take it. And, I had, I was starting to have myopathy, and I started taking lion's mane, and it made the pain a lot better. And so I run into these stories all the time. I just thought I'd mention that because that was literally today. So I think that's really funny. And when people say it made me feel better, it made my nerves better, or it helped with my nerve damage, I think that this mechanism here for neuroprotection and dendrite growth is probably what they're feeling, is the most obvious effects of the many effects that we're getting from lion's mane, and specifically Erinacine A. So you can see that what happens is Erinacine A actually acts on the cytoskeleton of the glial cell, and this stimulates the release of neurotrophic factors like nerve growth factor and other. You can see there's other cythine diterpenoids, and the cyhookerin C is actually another very potent compound, and it also it causes the release of BDNF as well, basal-derived neurotrophic factor, which is another important aspect of maintaining neuronal cellular health. And this goes on to protecting the neurons through neuronal cell death or apoptosis, neurite outgrowth to help new neurons form and grow, and axon regeneration. And the axon is what you can see, the yellow is that myelin sheathing we were talking about. So that's another really important aspect to these medicines. And this, we're now seeing, can even lead to positive psychosocial effects of these medicine, from these medicines. And there's a term, what we see in the literature, called Erinacine A enriched mycelium. And this just means that they standardize the amount of Erinacine A because it's the most commonly researched compound in the fungus. So they've standardized it to have like 5% in, at least 5% in the medicine that they're giving now to Alzheimer's patients in some fairly large trials in China. And we're seeing that the neuroprotective effects in Alzheimer's disease-related pathologies in mice is having significant results. And that's what led to the studies in humans. But we're also seeing that, and I want to back up to some of the other effects. The antioxidant effects are sort of a parallel effect to why it helps the nervous system, because like I was saying, inflammation on the nervous system is implicated in a lot of neuronal diseases, especially neurodegenerative diseases. So if you think about these two actions it's having of actually affecting nerve growth factor synthesis, but also reducing inflammation, and that in turn attenuates neurotoxicity, as we're seeing in this model of Parkinson's disease. And so we've also seen some pretty interesting studies coming out recently about the psychological effects and the social effects, because when, especially in neurodegenerative diseases, when you're declining, it's actually a positive feedback loop, not positive in the sense that it's good, but positive in the sense it keeps happening. When you have an affected quality of life and you're having memory problems and it affects your social life, it continues to affect your psychological health, which actually leads to a more rapid decline. And this is what we see in the decline of the brain in Alzheimer's patients in homes where they don't have family visiting and they don't have constant stimulation through language and touch and music. So that is sort of a way that we can treat the anxiety, the depression, and the other psychological effects and the psychosocial effects of these diseases. And we see this in a study where they ameliorated anxiety induced by continuous sleep disturbances. So if any of you out there ever have sleep disturbances, and that's a real thing, maybe consider reading up on this study and seeing if lion's mane in a regular dose might help. So now we're going to move on to the hypothesis of what we did and what we were looking at.
Yeah, so given the fact that Hericium erinaceus is known to contain these bioactive cythine diterpenoids, and Hericium abietis is so similar morphologically, ecologically, and phylogenetically, we figured there was a good probability of finding these erinaceins in Hericium abietis. As Jax mentioned earlier, there's a lot of these cythane diterpenoids found across other fungal species. So I think mentioned over 170 different of these cythane diterpenoids. And so we hypothesized that Erinacine A would be in the Hericium abietis in detectable amounts. Our experimental design, naturally start off with cultivation, increase the mycelial mass. So taking some samples of mycelium and getting them in our liquid culture to grow, letting them go for long enough to be able to start producing the secondary metabolites, because erinacines are secondary metabolites. They are not part of the initial mycelial growth. They have to be old enough really to start producing secondary metabolites or yeah, and then once we've got the mycelium grown enough, move to the extraction to separate our cythane diterpenoids out of the mycelial mass to be able to send off our extractions for liquid chromatography to detect for cythane diterpenoids. Cultivation process, everything that we were researching about the best way to do a liquid culture cultivation used bioreactors, which would be ideal since there's constant agitation going on. The mycelium is broken up and creates the maximal surface area for every little piece to be able to get nutrients from all around itself. It's also, bioreactors have a release for CO2 to be able to go out because mushrooms do respiration like animals do. They take in oxygen and release CO2. So there has to be a way for CO2 to get out and oxygen to get in. It's much less incubation time compared to solid state cultivation. Again, going back to the maximal surface area with the mycelial pieces being broken up in a liquid culture. And it's a fairly easy process to start, so long as everything is sterile. Just add spores or mycelium to the liquid medium to get it to start growing. Our cultivation method did not involve a bioreactor because we don't have one at the Evergreen State College. So as you can see in this photo, we have a temperature-controlled shaker bath with flasks in there and lids that are just loose enough to be able to wiggle to allow for CO2 and oxygen exchange. We had two different Hericium species, Hericium erinaceus and Hericium abietis. Took chunks of mycelium from agar plates with a little cork bore, put it into the flasks with our liquid culture medium. The liquid culture medium was a very simple recipe, just glucose for the main source of carbon and oxygen to be able to build chitinous cell walls for the mycelium. Peptone was the big source of nitrogen for DNA replication. DNA bases and RNA bases require nitrogen. Nitrogen is also very important for amino acids and enzymes, so we use peptone for that, as well as potassium phosphate for all of the other action potentials that need to happen, energy production, ATP. So very simple nutrient medium, glucose, peptone, and potassium phosphate. Our shaker baths had them set at the ideal temperature, and we had 3 time intervals that we pulled the flasks out to check for the secondary metabolites. The data that we were looking at suggested that the highest concentration of erinacine, a few of the papers were a little different. One was about 14 days, another one was about 21 days. So we did 3 time intervals over the course of 28 days to see if there was any difference in erinacine levels based on how long they had been going in the liquid culture.
So yes, the mycelium of the Hericium erinaceus and Hericium abietis are both suspected to produce erinacine-like compounds, which is the 5,6,7 tricyclic ring alkene hydrocarbon. They produce it in a metabolic pathway like many other hydrocarbons and many other compounds in biological organisms. They produce it over time, which means that they have specialized genetically produced from the genome enzymes, which take a molecule and using energetically active regions of this enzyme, it'll cleave these molecules, which facilitates reactions between molecules and builds the molecule that is either beneficial or neutral, or sometimes even detrimental to the organism. This being said, we took our time intervals and we would extract the Erlenmeyer flasks from the shaker baths at a particular time point. And because of the tricyclic ring hydrocarbon structure of Erinacine A and the other erinacines in the group, this makes it strongly or somewhat strongly nonpolar, which means that it's not actually, it's present in the broth, the cultivation broth, but is somewhat, it's actually separatable from water using chemistry methods which remove solvents by reductionistically solubilizing them in either polar or nonpolar solvents. So using the analytical methods that we planned in our experiment, we needed to remove Erinacine A from the polar water-based liquid broth that it was growing in. To do this, we use nonpolar solvents, just as the theory of the molecular differences would suggest. So here we can see a polarity scale, actually. Oh wait, actually, if we could go back, please. We can see water at the far right, which is strongly polar, and then you have cyclohexane, which is an alkane hydrocarbon, and that is strongly nonpolar. Between that, you have benzene, ethyl acetate, acetone, and then you go further towards polar as you increase the electronegativity differences within the molecule. Most hydrocarbons will have mostly carbon and hydrogen inside with less oxygens, which means that the electron distribution is somewhat even, and that generally facilitates a polarly acting compound. Yeah, so that being said, next slide, please. So we have our steps here. In our extraction, we would take our nonpolar, or sorry, our polar broth that we cultivated the mycelium in, and we froze it at a very low temperature in our lab facilities freezer farm room, just a very industrial, strong freezer room. We would freeze our samples in special jars, with perforated lids, which allowed for gas exchange. And then we would move those jars to the freeze dryer, which we had access to in our lab space. The freeze dryer would evaporate the water out of the frozen broth, which would leave only the sugars, peptides, and nonpolar molecules in theory left. If our target molecules were extremely volatile, then we would have a problem here because the vacuum pressure would cause its boiling point to lower, which would mean that our target molecules would evaporate from our broth, which there may be somewhat, there may have been some amount of this in our experiment as a part of error analysis, but because we used the same procedure for every sample, the amount in which this would happen would be somewhat even, and therefore our data would still be accurate. So that's step one, lyophilization or freeze drying. Step two, we would add 80% ethanol according to the strength of the polarity identity of our compound. 80% ethanol seems to be the most efficient at solubilizing Erinacine A in particular. That's based on the literature that we based our experiment from. The best results that other researchers got was with 80% ethanol. And so we were able to obtain 99% ethanol from our lab space and then dilute it down to 80% with deionized water. So we would add that to our freeze-dried culture broth, FBF or FDCB for short. And we would add it to a reflux condenser apparatus as seen on the right. And we refluxed it for a desired period of time that was remaining constant through the whole process. And this increased the temperature, of course, which solubilized the Erinacine A. It also separated the Erinacine A from the solids in our mixture. And it also would not solubilize the sugars or peptides in a mix, which is very important because now we're moving away from sugars, we're moving away from proteins, and we're selectively extracting our Erinacine A from the cultivation broth mix. So once the reflux was complete, we cooled it. We also added ethyl acetate and we cooled it somewhat and then evaporated the mixture with a vacuum apparatus, which is part of the flow hood as seen on the right. The vacuum would pull and evaporate the ethanol. Once we filtered it, I'm so sorry. we after reflux, we filtered the mixture through a vacuum filter. We separated the solids, like the sugars and peptides, from the liquids, which is mostly the ethanol, the water, and the target compound. And then we evaporated that to a low amount of liquid, which we added ethyl acetate to, which is even more strongly non-polar. And then we added water to that. So as you can see on the bottom middle partition block, the addition of water separates the leftover still present sugars and peptides. It separates them into the water layer. And then in theory, we have our non-polar target compounds trapped in the ethyl acetate layer. The ethyl acetate is on the bottom and the water is at the top. You can see that the ethyl acetate layer is much clearer than the water layer, and that's because there are caramelized sugars and solubilized peptides in the water layer above, which discolors the solution. The ethyl acetate layer has very small amounts of Erinacine A and other nonpolar compounds solubilized in it, which means that its net clarity is actually not really affected, so its color should be clear. After this, we separated a little bit of we separated the partitions using the separation funnel. And then we evaporated we added sodium sulfate, which is a hydrophilic salt. And that basically pulls the last of the water out of our solution. And then we filtered that out, separated the liquid layer from the solid salts, and then evaporated them, which left us with what we call crude extract, which is mostly non-polar compounds, which are ready to be tested using LCMS. Next slide, please. So what is LCMS? This is the instrumentation that we decided to use on our analysis section. LCMS stands for liquid chromatography mass spectroscopy. And it is ideal along with a couple other analytical methods, for elucidating and identifying particular nonpolar large molecules. Something like GCMS, which is gas chromatography mass spectrometry, is not suitable for large compounds, especially ones with like substituent parts, because it is dangerous for the machine to run, and it's not an optimized piece of technology for that application. So we use liquid chromatography mass spectrometry. So what that means is we injected our sample into one end of the machine, which heats it through something called a column, which is very thin tube. It heats it at a steady increasing rate over time. And then it holds at a certain temperature, and the molecules of a particular boiling point will pass through at particular times. So that leads to the second part of the apparatus, in which the mass spectrometer portion will analyze the signals which result. So the signals are coming from this part on the right, as we see the enlarged block there. There is a current which is facilitated using magnets, electromagnetic energy, to energize the molecules which are passing through the column. And so, of course, they're coming through at a particular time, which is identified later. And as they pass through this part of the instrument, they're energized and they fragment. And that fragmentation will break the molecule into ionized components. And so what happens there is that you have like smaller portions of your molecule breaking off and then passing through this part, hitting the sensor at the back. And based on the retention time, the signal, which is produced by the contact of the ion with the sensor, and then how many different portions, how many different ions hit the sensor, you can use this to back engineer the molecule, which theoretically has passed through your instrument. Essentially, you are looking at the characteristics of the building blocks, the arms, legs, torso, head, and other parts of your molecule, figuratively speaking. So we used our mass spectrometer in negative ion mode, which means that we were identifying the negative substituents of our molecule. With the help of Anna from University of Washington, or was it Washington State? Yes. Yes. So let me just be absolutely clear on that.
Washington State. Yeah,
Washington
WSU.
State.
Yeah.
WSU. Thank you, Dax. With the help of her, we are very thankful to have access to this instrument at a student feasible price. So thanks again to her. And next slide.
All right. Okay. So we just wanted to give a brief overview of the differences between chromatogram and mass spec, since they look very similar to each other. I was very confused at first. Took me a while to get it, but this will make our results make a bit more sense. And it's also just very useful to know because it's heavily used for verifying the presence of compounds. And so here on the left is the chromatogram, and it is showing the presence of molecules that are very, very close to the weight of the target weight. And so each one of the peaks here in the left chart are represent a different molecule, and the height of that peak represents relative abundance of that molecule. And on the right, we have the mass spec. And what this is doing is taking one of the peaks from the chromatogram. In this case, it's taking the 5.32, the biggest peak, and it's showing how that molecule, like Aiden was saying, breaks apart or fragments. And what is really cool about this is that specific molecules have a distinct fragmentation pattern. And so what you do is you get a standard, which is essentially a highly pure sample of the compound you are looking for, and you run it through identical machinery and conditions, and it will produce that molecule's fragmentation pattern. Then you can take that fragmentation pattern, compare it to the fragmentation pattern of the molecules in your sample, and then that will, if they match, then that is then they are likely the same molecule. And something to note is that molecules that are the same weight will break down differently, because even though they're the same weight, there are still differences in the molecular structures. And I stress the importance of using the same equipment and conditions, because there's many types of chromatography and conditions that can be changed. And having everything the same is crucial for finding a match. And unfortunately, the standard for Erinacine A costs $35,000. And apparently that's not in the school's project's budget. Crazy, right? And so we were unable to get a confirmation on the presence of Erinacine A, at least yet. And that should be it for this one. So with that, let's take a look at our results. So we targeted the math for Erinacine A, which is that 431.2433 right there. And there's a little bit of wiggle room for the weight of compounds that can make good candidates. So here we have a chart generated from a software called Neurogenesis, and this shows 9 possible candidates that fit within that range. And we narrowed that down to 3 likely candidates when matching the retention time, which is the number on the left. And that's essentially the time it takes to move through the column, as Aiden was explaining, with the chromatograms peaks. Next one. So yeah, these next 3 slides are the chromatograms for each of the 3 mushrooms we grew at each of the different time intervals. And this slide is looking at Hericium erinaceus. And what's cool is you can see the differences in concentrations and composition of different molecules over time. You can see them peak, decrease, get lower. And when I'm going through these, we will see that the peaks at 5.32, which is the big peak right there, and 597, which is just to the right, are in all of our samples and all of the time intervals, which makes it an excellent candidate for possibly being Erinacine A. And so this one is just the first specimen of Hericium abietis. And we will explain why there's a little question mark next to Hericium abietis later. And like I mentioned, you can see those 2 peaks in all of the time intervals, and it has a few differences in composition from the previous slides. And then the last one, there it is again, the second sample of Hericium abietis, and there are those same peaks present as well. And again, it has a slightly different composition than the other 2. And we just wanted to show this graph to help illustrate how we visualize and analyze the data. This is looking at that big peak, the 5.32, which was the most prominent peak in all the specimens, and showing how the abundance of that molecule changed over time. For Hericium erinaceus and the first abietis specimen, the abundance of this compound peaked at the second time interval and then began decreasing, excuse me. And for the second abietis specimen, it had the highest abundance at the last time interval, which is just interesting and raises some questions.
So, actually, excuse me for a moment. They're running the loud piece of instrument.
Here, I'll just
No,
okay,
I'm good. Okay. So, due to a lack of public information, public availability of data on genome sequences of Hericium abietis, we ran PCR, polymerase chain reaction, which is a genetic process for identifying fungi and other organisms. Any organism can undergo PCR, but from what we know. But because of lack of the availability of this knowledge on abietis, we were not able to 100% confirm the identity of Hericium abietis, though one of our classmates during our time of experimentation, our classmate grew the same sample which we tested for our experiment. The same sample was grown on conifer wood, which is an exclusive trait of the Hericium abietis within these two species. Erinaceus is not seen to grow on conifer. So we were able to see this other sample all the way to its fruiting stage, which morphologically resembled the Hericium abietis that we knew. So there's some conflicting data which we need to keep looking at, and we would encourage the public and other scientists to provide more PCR information or particularly full genome sequences, as well as ITS region sequences to the public, particularly the GenBank public library of genomic information. If that was more available, then the ratio of information on Hericium abietis and Hericium erinaceus would be more equalized, should allow us to further specialize our identification strategy on Hericium abietis. Though we tried, we were not able to get conclusive distinction between the two because our primers did not have the appropriate differences which would, isolate their identifiable traits, their genome regions of traits. So another big problem that we ran into was, of course, as my entire team has mentioned before, the lack of availability of an Erinacine A standard within a reasonable price range for researchers. So if this was more available, then we would be able to afford a sample, which we could run through our instrumentation and get a better idea of what the data for Erinacine A fragmentation through LCMS would look like. And then we can look back at our results and get a better confirmation on the presence distinctly of Erinacine A within our samples. Next slide.
Implications and conclusions for this project are pretty exciting. If we can keep further confirming that Hericium abietis is a potential source of medicine, it could potentially be much easier and more widespread to be able to cultivate and get out to people, since conifer substrate is more abundant and affordable than hardwood substrate, which Hericium erinaceus prefers. There's also the potential of finding novel bioactive compounds in Hericium abietis that, again, would take a lot more research, breaking down the extracts even further, doing thin layer chromatography, TLC, to separate out different molecules, and then NMR to be able to actually build those molecules to see exactly what they are. There's a lot of potential in this, and we hope to keep continuing looking at our data and to encourage other people to run some experiments on Hericium abietis as well.
Nice. And that's it.
We're done.
Yeah, thanks.
Let's move forward to the acknowledgements. The references.
Oh, yeah, that's right. The next slide. Where's our, I don't think our acknowledgements are on here. Well,
No, I didn't mention them
we
at the
acknowledged in
beginning.
the beginning.
Yeah. But we also have references if anyone would like, just get a hold of us and we can point you in the direction of kind of where we went. And I'd like to echo what Zachary said, that although it like what we got is a partial fingerprint, right? And we can't confirm it. And also we can't confirm a fingerprint in the database. So first we need to get a good print in the database, or a standard. And we could do that just by separating out our sample and getting a fraction of each molecule, and then testing those independently through like nuclear magnetic resonance or X-ray crystallography. Because when you're confirming a molecule, you really want to use multiple multiple avenues of instrumentation and analysis. And so that's typically what's done in the literature. We thought it might be easier to obtain a standard. It wasn't, but we could make our own. And then once we did that, we'd have our fingerprint. Then we could, anything we can put in our LCMS is now a match or not. Either the fingerprint is matching what's in the database or it's not. And so that's kind of, yeah. So we look forward to continuing that research. And we actually do have NMR at the Evergreen State College. So it's totally within our reach to continue this and give us the other half of that blueprint.
Other good news is I'm sure we can pay for the standard on the cambium front as well, so we can continue the research with the UP HPLCs and just actually getting that stuff calibrated over in Michigan.
Yeah. So there's a potential. Yeah, there's extract analysis of mushrooms going on in Michigan now that has the potential to help us get those standards. And actually, we heard rumor of a couple companies in Japan that have affordable standards for Erinacine A, but I think it's just being able to find them and communicate effectively to these research companies that are right now just a rumor. But yeah, and we'd like to now open it up for a questions and answers section. If anyone has any questions about our research or just about Lionsmane or the Hericium genus in general, we can do our best to answer whatever question comes our way.
I've got a question. So do you suspect that the Erinacine is again primarily in the mycelium of the abietis as opposed to the fruiting body? Or do you have any reason to suspect one over the other?
Yes, all the most current literature is showing that the cythane diterpenoids are only found in mycelium, but it's curious that there are a couple of papers I would say, I don't remember the exact dates, but I want to say at least 15 years ago that found apparently erinacines in fruit bodies, but that was also looked at afterwards for confirmation and no one could find it. So, and also, yeah, there's a few, yeah, it's like that when you're hunting through, and there's thousands and thousands of peer-reviewed published papers on Lionsmane now and its compounds. It's extremely popular research fungi in China in particular, but now expanding through Eastern Europe, and even a few in Mexico and I think the United States, but it's really limited. Our medicinal mushroom research is not very funded in the United States compared to particularly China, and their natural products research sector is just much bigger in general.
Right on. Well, thanks for the presentation.
If I may chime. Yes.
Yeah. Anyone else have a?
I have one thing to add to that. It's also very common that in cultivation of Hericium, you have fruiting bodies which are small and closely hug the substrate, which means that when they're taken off of the fruiting substrate, if you're analyzing the fruiting body, it's very easy to get some of the mycelium mixed into that. So it's totally up to the exact protocol of the researchers. And then it's also possible that, maybe as the mushroom grew, it sort of blew out some of its own substrate into the fruiting body, which it sort of folds inside of itself. So those are a couple factors to consider if you do see. Yeah,
that's a very good point, Aiden. That's a very good point, Aiden. And it can work the other way as well, because they've also found hericinones in mycelium, which then that was not followed up when it was investigated. And I've cultivated Lionsmane for many years, and the way that the fruit bodies actually flatten and grow against the substrate sometimes look like mycelium and maybe could get mixed in depending on the method. Is it liquid culture? Is it solid culture? So yeah, excellent point.
Another quick thing to add is in terms of the presence in fruiting body versus substrate argument. It's a very controversial topic in the entire field of supplements, medicinal mushroom marketing. is that it's possible that Hericium is producing cythine diterpenoids and hericinoids for different purposes depending on where they are in the fungal structure, because generally when you have a biomolecule that's formed, or even an enzyme that's formed, or a protein in any organism, there's a role that it plays oftentimes. Otherwise, it's not energetically favorable for that substance to exist. Therefore, both of these compounds probably play a role in the life cycle, but that is not like confirmed of like what role that plays. Within our fungal kingdom course, we discussed this with faculty briefly, and one of the ideas, which is just an idea, not supported by evidence, but one of the ideas that we mentioned at one point with faculty in conversation was that it's possible that cythine diterpenoids are produced within the substrate as a defense against the tree's immune system, because oftentimes you'll find Hericium fungus growing in dying trees. They're not completely dead yet. They will be present in dead trees as well, but oftentimes standing, dying trees, sometimes standing dead trees. So it's possible that it's some sort of chemical warfare, if you will, against the host organism. It could also be somewhat of like a preservation strategy, maybe to keep its territory safe from other bacteria. Those are just ideas, so not supported, but yeah.
Yeah, fungi are definitely well known for their defenses, right? Chemical defenses to even create boundaries with their chemistry. And you can see these zones in petri dishes that form, and with yeasts especially. And that's where we get antibodies, is in those zones, right, of demarcation. So yeah, that happens in trees as well. You can cut into trees and see this.
Absolutely.
I have a question related to the compounds you guys have been studying. Do you think that there's evidence to suggest that these compounds could be found in possibly other species of fungi, or perhaps plants, or bacterial colonies in significant quantities?
So speaking to the cythine diterpenoids, that specific 7,6,5 tricyclic ring structure, that from everything we looked at is only found in basidiomycetes or higher fungi, not even in yeast, or it seems to be part of their secondary metabolism system in white rot fungi, but it's not just found in the Hericium genus. For example, the Cythis genus was where they were first discovered. So really, we couldn't find anything in bacterial or plant or lichen or even yeast. It's really only seems to be basidiomycete, the sort of, what we think of as cap and gill mushrooms, but obviously they're not all caps and gills as we see. They can have spines and lots of different shapes, but yeah, that's and I don't remember if that was really been investigated at all as to why that is. There's a few other tricyclic ring structures that have high bioactivity. I almost put a slide in about the other ones just to show, and there's some found in bacteria as well. They're not the cythine backbone, but it's kind of similar. But the point is those tricyclic ring structures seem to have high bioactivity. And if we go back to the slide here, which one is it? I can illustrate the point here, is that you can see that although on the bottom, the cyhookerine and at the top of the list and the Erinacine A, and that's showing that it's going to a positive, and then you see compounds that are going to a negative above that. That's showing the effects it has. So actually at the top, scabronine and this compound 15, which I believe is either unnamed, or there's also, they were talking about one that was synthesized, but it actually has a negative effect on the release of neurotrophic factors. And that can be good because, I think often when we think like neuronal growth, we think good, but there's a whole homeostasis to neurons, and they need to die and regrow at specific timings, and circuits need to be held back from overgrowth. Or overextension of the neuronal net can lead to hypersensitivity of the nervous system. This is implicated in such things as autism, and so it's important to recognize that neuroplasticity should always be balanced with neurostability. So it's not always like I make my brain more neuroplastic, things get better. Things can actually get worse psychologically if you're so neuroplastic that you can't handle a shock, for example. And shocks to our nervous system happen by surprise and fairly often, whether we like it or not. So you can see that it could be potential benefit of some of these compounds with this 5-6-7 tricyclic ring structure to even have a negative effect on neurite growth for people who have maybe too much neuroplasticity, for example, autism or other hypersensitive, nervous system disorders.
Well said, bro.
And that was excellent.
It's very interesting.
Yeah,
I just wanted to add there. Oh,
yeah.
I just thought this was kind of funny, but I've heard many times, and I'm sure my teammates as well and other mycologists, when it gets to the topic of genes and mycology and genetic modification in particular, a joke is often made that these medicinal compounds in fungi, which are eukaryotic organisms like humans, that the genes for the production of things like Erinacine A, for example, or more commonly joked about is psilocybin. People will jest about using some tools like CRISPR or such to edit the genes for the production of these medicinal or psychoactive sometimes compounds into themselves. And that's not necessarily a good idea, of course, for many reasons, but one of which is, as Jax mentioned, that too much of a good thing is a bad thing, and when you have too much of the activity of even something that we see as medicinal, like Erinacine A, from the studies that we've seen so far, it could be very damaging. I'm sorry, I just got a call. So, anyways, that was the end of my point there. So I just thought it was funny.
Thank you to the team. It was really, really
Jax, you're
interesting.
muted. Thank you, Marge.
Oh, yeah, thank you.
Yeah. Yeah.
Alan,
Seen you guys
Rockefeller.
before. You got to come out again. You have to come out and visit again.
Yes, I would love to come visit again. I'd love to come visit again. But Alan, I would love to hear any input you had, or I thought I saw that you unmuted for a second there.
Yeah, this is really cool. It would be really neat to test the fruit bodies as well and just see like the relative abundance of the ericinines in the fruit bodies versus the mycelium. And I'm also really curious, like between the different species of Hericium or different collections of the same species, it'd be really interesting to see how much it varies. And how much these chemicals vary depending on different substrates that you feed them in liquid culture. And I know I offered to look at these DNA sequences, and I've been traveling for the last couple of months and I haven't had a chance to do that, but I would still like to get everyone on a Zoom call and just take a look at the DNA sequence data and see if I can pull any useful information out of that.
That would be awesome.
Yeah, that would be great.
Yeah, thank you so much for that offer. We really appreciate that.
Awesome.
Yeah, and I imagine it's not a good time now just because you're all probably tired from this, but if you did want to do it now, I'm just sitting here at the computer with the internet, so I could do that in a few minutes or whatever, but some other day is fine too.
We should just get it done.
I can shoot that over, or I guess we all have the access. It's all on the SharePoint too, but we can shoot that over just all the data files from PCR over to you, Alan, so that you can take a look. And what do you guys think? Do we have a are you guys down to take a look at it tonight, or should we schedule it another day?
I have company, so unfortunately I can't today, but you don't have to wait up. I'm also.
Oh no, we have to wait for you, Jax. It's a necessity.
Of course.
Well, I'm
Yeah, I
I'm
think
Yeah,
we
I'm
need.
done.
So speaking to your last point, Alan, there's been quite a few good papers that have done nutrient differences and its effects on specifically erinacines and Erinacine A production, both in solid state and liquid culture. But your other and we kind of modeled off one that had optimized that for high Erinacine A production, because we were really trying to optimize it for multiple species. But like you're saying, I with the variation in different species or even different collections, I haven't seen any of that, and I think that could be really significant, because wild type specimens are going to have a different nutrient broth, so to speak, than something in our petri dish or a culture flask. So yeah, I'd really like to see wild type versus cultivated, and then have a few different variations of nutrients on the cultivated end, and then a few different wild type collections, together. And
Yeah, absolutely. And it'd be cool to take the spores from some of the most potent ones and maybe make monokaryons and crossbreed them and see if you can get something that's even a little bit stronger.
yeah, start optimizing the growth for medicinal compounds rather than the look or the taste. Yeah.
Oh yeah.
One thing I was wondering is, which solvent do you think is best to extract the ericinines? And do you have any insight into the thermal or pH stability of those compounds?
Yes, so we chose ethyl acetate because it wasn't extreme, like it wasn't notably extremely polar. And Erinacine A was seen to be extracted using ethyl acetate and ethanol diluted to 80% as the most optimized option based on literature that we saw about optimization of extraction. So yeah, we've really studied that extraction process in the literature pretty heavily, and some of the papers within the same study would use different concentrations of these solvents, and then they would, on the same original sample that was grown in the same conditions and everything, and then the results would be different based on the concentrations of solvents that they used. I'm particularly thinking about one article that I used, but we also saw the same use of ethyl acetate, the same use of the relative dilution of ethanol in multiple papers. Oh, and the thermal stability seemed to be pretty stable. That's why we felt that it was safe to undergo reflux.
Yeah, reflux is kind of standard,
cool.
and I would say ethyl acetate, being that unless you're going to recapture it, it's going to be expensive and dangerous to work with. So ethanol, yeah, the 80% ethanol seems to be it's maybe not quite as good, because you're also getting all the polar compounds. But I think that's the thing that people in the supplement market mostly try to push on their products, I've seen, is that it's neuroprotective or neuroregenerative. But a lot of times it's just the fruit body and not the mycelium. And so a lot of people point to the Erinacine literature that's only found in the mycelium, but then there's a product that's just a fruit body. So the literature on the tested supplements, because there are some papers where people just are testing supplements out there, there's pretty wide variation on its effect and neuroprotective effects. And obviously there's a lot of variables when you start talking about a supermarket supplement or something. But it'd be nice to see more standardizations, I guess, is my point. And so that people can say, yes, I have X amount of hericinones in my product, and it should be, based on literature, producing such and such effect.
Yeah, that'd be really cool if that was in the, just listed on the bottle. And hopefully in the future that will be. Also, I don't know if anyone really has any idea like how much of these ericinones should be consumed by a human in order to get the effects.
That's a good point. I don't have the dose on hand because we were mostly going with Erinacine A. But I do have it here somewhere. Let me see if I can pull it up real quick. If you guys want to, I can circle back to you. Let me see if I can get that.